attendees, sirs and madams, and our dear students and respected speaker, Dr. Ashish Ji. So, as you know, we are conducting this online free webinar under this Science Dialogue series. This is free, means this is not under banner of any registered organization. We are just only trying to disseminate knowledge, taking help from expert people, so that knowledge will percolate and overall knowledge pool of our society will improve. Because we have seen that certificate holders are many times more than the actual deserving, or you can say those who are knowing this subject. So we cannot change the system, but we can try to improve overall standard of bioscience subjects. So I'm thankful to speakers who are willingly joining us, and they are delivering his or her lecture. And we are not also randomly picking the people only based on their degree. We are picking the people, resource person who has good credential. You can see all this is seventh lecture, and we have other in queue. So all have very good credential. They have taught their teaching credential is good, or they have very good background either from Indian Institute of Science or Max Planck and Oxford. That type of people are delivering, so that quality of education will improve. And nowadays it's easy, only we have to invest time. There is no cost involved, just only making flyer and other things. So we take help from other person. So since we need only internet and laptop to disseminate knowledge, and so many people are willing to interact with their student and other research scholars. In this way, this is going on, and my target is to conduct this type of more than 1,000 up to 1,000 lectures, so that overall knowledge pool will increase. And those who are knowing me, so I am very passionate. I have conducted 26 hours of online recombinant DNA technology course, and each course duration is more than 30 hours. And eighth batch of genomics and proteomics course is going on. Means I have along with some 2 or 4 hours of engagement of other teachers in these courses. So I have delivered more than thousands hours of lecture in addition to my regular teaching in universities in this corona. So with this introduction of this campaign, I now happy to introduce our today's speaker, Dr. Ashish Warghane. He is working at Gujarat Technological University, and his background is also good because I have called him in my GSBTM sponsored crash course. So he showed very good impression on the student because that was GSBTM sponsored, and participants were from different colleges of the South Gujarat region. So this is brief introduction. Remaining thing, either he himself will say you, or from his teaching you will understand. And he's in good universities of the Gujarat Technological University, and I have interacted with his student also. I have taken feedback from his student also, because my penetration is all over Gujarat. In means students are in all the universities of the Gujarat and outside Gujarat also. So I take feedback from students also, and his background is also. So with this, I welcome Dr. Ashish Ji, and I'm thankful to him for sparing his time for disseminating the knowledge to increasing overall all pool of the knowledge. Because I always observe and convey to my student that combined population of America and Europe is less than the our India population, but still we are dependent on for scientific thing. We are dependent on European country and American country, because we have huge population, but we have less skilled people. We have so many degree holders people, but overall knowledge pool is not that much. So we are trying our bit. So yes, welcome Ashish ji. Please unmute your mic and show your presentation and discuss. And attendees, I request you to interrupt him in any time whenever you find difficulty, doubt, so you can raise hand and ask question to him. This is not one-way interaction. This is live lecture. I can give you recorded lecture also, but I think live lecture is more interactive, two-way interactive, so that participants can ask question, can discuss, debate. In this way, knowledge will improve. Otherwise, recorded lectures are available, but sometimes suppose you are stuck on some terminology, no one is there to help you. So here live interaction have more opportunity to interact, and you can also show your talent through your good questions. That also, so I know Ashish ji because he has also attended one of my RDT course. So I know he is better than me or sometimes equivalent to me. So I called him as a resource person because here in online mode I don't know your background, so many participants background. So you can interact, you can ask puzzled question, good questions. So it will also give your miss, it will give us opportunity to judge your strength. So you must interact with teacher, you must respect the teacher, but don't be like obedient, that much obedient कि sir जो बोल रहे हैं वही सही है 100%, then science will not improve. You must respect, you must moderately ask question, you must debate. Yes, Ashish sir.
Thank you. Thank you, sir. Thank you very much. And you are taking a very good initiative to connect the peoples. And I'm really passionate about your teaching, and you are encouraging the other students how to do the research, how to do the academics. No? Thank you very much for giving me a chance to deliver my talk here. And sir, now I am start to deliver my lecture.
Present now नीचे.
Yes, sir. So my screen is visible, sir.
Yes, yes, yes. तो स्लाइड मोड में कर लीजिए.
Yeah. Yeah. So now it is okay.
Yes,
Now it is okay, sir.
yes, yes, it's okay.
Thank you.
But
So,
attendees दिख रहा है ना आप सबको?
हाँ यस
Yes,
सर
sir.
यस सर
Yes,
Good,
sir.
good.
so almost we are reaching to good afternoon to good evening now. So welcome to all into the lecture of viruses. And intentionally, I put the title of this lecture is that viruses, the unsolved question. Still, we are finding the remedy for the viruses. We don't have any good remedy with us. We have only the what you can say immune boosters with that, which can kill or which can inhibit the virus replication. But still, we are searching the so many answer regarding the viruses. So, as told by the Dr. Gopal, it is an interactive session. You can stop me anywhere else. So let's start. So, what is viruses? Before starting my lecture, I'm asking the one question you that. How many viruses are present in the world? Can anyone tell me? Yes, audience, please. How many viruses? I'm asking in number.
These
So,
are uncountable.
yeah, right. Just which I mentioned here, more than star present is about 31. These are the number of viruses is most ubiquitous. So we will start with the introduction first. So very simple definition of the viruses as our class having the some student and some professional also. So I'm going in a very similar fashion. So viruses is a intracellular obligatory parasite. Means वायरस जो रहेगा वो सेल बहुत बार पूछा जाता है वायरस लिविंग है कि नॉन लिविंग है राइट जैसे कि वो सेल के अंदर में लिविंग है सेल के बाहर में नॉन लिविंग इट्स द सिंपल डेफिनेशन इज इंट्रासेलुलर obligatory parasite then it is very small in size mostly the nanometers not seen by the naked eyes. You must have an electron microscope to see the virus. More than 2000 plant viral diseases known in the world. And I'm very happy to tell you that from last 12 years, I'm working on the viruses and mostly on the plant viruses. From that, I got to know that the first discovery of viruses goes through plants only. That is tobacco mosaic virus, right? Eventually, it is infected to the humans also. but the discovery goes to the plant species. Then most of plant viruses, when we are looking, they are the RNA genome. Till date, we don't have any magic bullet available to control the viruses. Still we are searching. Hence the title is like this. Till date, only 5,000 viruses identify the researcher or scientist, and this gamble or this study is still going on. Where once we are doing the more study and more study, we will get the more number of viruses. Why I'm saying it's gamble? Because so many times we are in a lose in this connection. No win-win situation is here. So general properties we will see, it is like very small, mainly 20 to 300 nanometer in size. From this figure, you can understand how small is the virus. If you see this bigger round, this is the staphylococcus. ये देखो ये staphylococcus है. Then it is a bacterium. Then उसके बाद आपको क्लैमिडिया दिखेगा. Then पॉक्स वायरस यहाँ से शुरू होता है. आप साइज से कंपैरिजन कीजिए कि वायरस बैक्टीरिया से भी कितना छोटा है. If the average size होता है बैक्टीरिया का कितना 2 माइक्रोन या 2 माइक्रोमीटर, एंड यू सी द अपना पोलियो वायरस जो है ना वो अराउंड नैनोमीटर्स में आता है. तो इट इज वेरी स्मॉल. आरबीसी भी देखेंगे 7500 नैनोमीटर है आपका. वायरसेस आर वेरी वेरी स्मॉल. Hence, it is one of the most difficult tasks to work with the virus. They are not culturable, right? If you want to culture, you require the live host, right? In animal cell, you can culture it, right? But in the case of again plant, you have to maintain the complete plant to culture the virus. It is not just like a nutrient plate.
Nims Vijayvara, do you want to ask some question or just by mistake you have raised your hand? Hello. Chandra Prakash ji, please mute your mic. Okay, so please continue.
Thank you, sir. So, if you are looking for the genome, the plant, these viruses either have a DNA or RNA. This is not case with us or plant. We having the both DNA also, we have the RNA also. But this is the particular thing in a viruses. Either they have DNA or RNA. This is the case of viruses. Then they are metabolically inert. They don't have protein synthesis machinery. They all time depend on whom? They are depend on host to take the fruits from the host. They don't have proper nucleus, cytoplasm, mitochondria. Just the nanometer molecule are able to kill millions of lives on a global. We've seen in a case of coronavirus. This is the importance of the viruses. No metabolic activity, then replicate only inside the living cell, not in inanimate media. This is the meaning of the culture. We can cultivate bacteria on petri plates, nutrient agar. You can't go with this also, right? It requires a living medium. Then, if you goes for the history of viruses, the first time you see the all one by year by year. These are the Adolf Meyer. First time in 1886, he observed the symptoms on tobacco plant. That is a tobacco mosaic plant, right? Sorry, tobacco plant. He observed, if you see in my cursor, he observed the symptoms like this. Sometimes it's pale yellow white against the green background. So he observed that yes, this may be a viral disease. Then Dmitry Ivanovsky. These guys, what they did, they filtered the sap of potato, right, from the bacterial filter. And that filtrate he sprayed on the healthy tomato plant and observed that the same disease occur on a healthy tomato plant. Means something is there which is transferred from 0.22 micron filter. Means this is not bacteria, which is bacteria is more size than this 0.22 micrometer. Then your fungi is not, but only one molecule can cross this filter, that is a virus. That study is did by the Ivan Moss, that is the Benjeric, and he did a very systematic study protozoa, plants, animal. In that case, virus is very, very complex in its genetic nature also. If you are looking for the capsid means a protein coat which is surrounding the genome that gives the particular symmetry of particular virus, right? And this symmetry जो प्रोटीन इसके ऊपर रहते हैं, जो उसके रिसेप्टर वो बनाएगा, then later on it is used for the host पैथोजन जो इंटरेक्शन रहता है वायरस होस्ट इंटरेक्शंस, उसमें वो कैप्सिड के रिसेप्टर यूज किया जाएंगे. This capsid is also facilitate the transfer of viral nucleic acid into the host cells. Again, viral genome is protected due to this capsid. Now, what is capsomere? The structural unit of the capsid. Capsid is made up of so many proteins, right? That single protein is known as capsomere. It is capsomere is a monomer of capsid, you can say. Then this is going to very important. What is the biological property of the virus, right? All viruses should have their natural host range. Now, what is the host range? One virus can infect so many plants, right? They have a particular host, right? Then mode of transmission is nature. Already in a nature, it is established. There are so many theories for the evolution of virus. Through nature, it is established that how the virus is transferred from one plant to another plant, one person to another person via particular vectors or by a physical mode of transmission. Then it is have a vector relationship. If we see that our dengue virus, which is dengue generally transferred by the mosquito like this. Herpes is also same. Smallpox also same. Then they have a pathogenicity associated with the disease. One particular symptoms you are getting once you are infected by the viruses. It shows some pathogenicity. Again, some viruses are able to infect only some tissues, not in entire body also. So such type of viruses also we have. These are the beautiful symptoms. This is the mosaic, real mosaic symptoms. If you see such symptoms on a mosaic plant, clear cut is that there is a TMV is there, tobacco mosaic virus. Then, now how it is make the infection to the us? This is the basically a lipoprotein surrounding the capsid in some viruses. If you find this is the virus. This virus having this receptor, right? Or surface marker we can say, and our host means your body having this kind of the receptor. So they have the perfect complementary to each other, and this way they can enter into our body, right? So now scientists is working on these virus host interactions. So from these only we will get the antiviral molecule or anti-inhibitory molecule, or even we can find the good candidate for the vaccine development in this area. So many viruses host cell as well as viral origin, then virus encoded glycoprotein, surface envelope. Most of the human viruses are helical in nature. Now what is the newly viral particle arise from the infected cell, right? एक वायरस जाएगा एक सेल में इन्फेक्शन करेगा और वहां पे वो रेप्लिकेशन करेगा. रेप्लिकेशन के बाद क्या होगा, वो सेल को ब्रस्ट कर देगा. अब जहां से वो जो वायरस निकलेंगे वहां से जो हाईली इन्फेक्टिव रहेंगे उनको बोलेंगे हम वीरॉन्स. These are the terminologies. Now you will have a more interested will be here. Now see, on these axis the blue line I am showing the property.
Like आप पॉइंटर को वो कर लीजिए ना आशीष जी, पॉइंटर से तो दिखेगा अच्छा से. यहाँ हाँ
दिस वन है सर.
हाँ देखिए, हाँ वहाँ पर पॉइंटर होगा. हाँ हाँ लेजर पॉइंट. यस, नाउ इट्स मोर विजिबल. यस,
अभी ये देखिए आपको ये ब्लू में जो दिख रहा है, these are the properties of the virus. एंड इधर देखेंगे आप बैक्टीरिया. ठीक है, अभी देखिए ग्रोथ ऑन आर्टिफिशियल मीडिया. इन द केस ऑफ बैक्टीरिया एस, माइकोप्लाज्मा एस, रिकेट्शिया, क्लेमिडिया वायरसेस में नो, राइट. डिवीजन बाय बाइनरी फिजिस सब में एसएस है. वाइदर दे मच हैव म्यूरामिक एसिड. एस है आपका बैक्टीरिया में, बाकी में नो, और एस है रिकेट्शिया में. अगेन दे आर सेंसिटिव टू एंटीबायोटिक. बाकी सब में एस, बाद में नो. So from this simple table you can understand how different is a virus. and how difficult it is a virus. ठीक है, बाकी सब लोगों में देखोगे आप 90% प्रॉपर्टीज आर सेम, बट ये जो बंदा है ना वायरस, ये अलग ही है, and hence it is a very challenges to work on the viruses. Apart from that, you will be have a more challenges when you will work on the RNA virus, right? अब मैंने यहाँ पे एक स्टार मार्क दिया था, राइट? इकलौता ऐसा वायरस का फैमिली है दैट इज अरेना वायरस, जिस वायरस में आपको राइबोसोम मिलेगा, पर ये राइबोसोम ये उसका खुद का नहीं है। जब प्रोटीन पैकेजिंग जब होता है ना, जब प्रोटीन का वायरस रेप्लिकेशन करता है और फिर कैप्सिड के अंदर में उसका जीनोम जाता है, उसी दौरान होस्ट का एक राइबोसोम का यूनिट उसके अंदर चला गया, hence that ribosome they have, but it is not in its own nature. That is, I am giving the star mark. From this sample table, you will got to understand how difficult is the virus and how it is different from the other organisms. So, then classification of viruses. This is the most basic task in the case of viruses. We generally for plant, those viruses infecting plant, we known as plant viruses. जो ह्यूमन को करेगा उसको ह्यूमंस, जो सपोज काऊ को करेगा काऊ वायरस, देन अगेन जो संतरे के वायरस को करेगा, संतरे के प्लांट को इन्फेक्ट करेगा उसको सिट्रस वायरसेस। ये बहुत सारा कॉम्प्लेक्स था, देन ट्राइवियल सिस्टम था, बट देयर वाज अ मैन नोन एज अ बाल्टीमोर who classify the viruses very particularly, and later on we are giving the nomenclature in that way also, right? In ICTV we are also giving our inputs for the classification and regarding the other viruses also, right? So based on the presence of nucleic acid, right? DNA RNA है या सिंगल स्टैंडर्ड है, डबल स्टैंडर्ड है, या फिर पॉजिटिव स्टैंड है, नेगेटिव स्टैंड है, या फिर डबल स्टैंडर्ड डीएनए आरटी है, उसके बेस पे वायरसेस को हमने कैरेक्टराइज किया है 8 डिफरेंट क्लासेस में, क्लास 1 से लेकर क्लास 7 तक, just like those viruses having double standard DNA, right? Those viruses having single standard DNA, then it become a double standard DNA like this, और जो उसका mRNA कोट होगा. So on the basis of presence of what kind of the nucleic acid in it, we into the 8 different category classes, and this discovery is goes to Baltimore. He gives wonderful classifications ones, and it is itself known as Baltimore classification of the viruses. And this is the guy. Later on, if you want to cultivate virus, you require the host, right? So what kind of the host we are using here? We are using the eggs, right? And these are embryonated eggs, means the living embryo present inside the egg, which we can use as a host for the cultivation of the viruses. One more thing, there is a no universal cell that can support the all viruses. In our, we have the one common media that is a nutrient agar. Most of the bacteria उस पे grow होते हैं, ऐसे हम बोलते हैं, ठीक है? But ऐसा कोई चीज viruses में नहीं है, right? So it requires a specific host. Virus tend to be host specific, thereby human viruses grow best in a cell of human origin, not on the others. But our very close is that mouse. So we can use the mouse, guinea pigs we are using. Then therefore it is in a laboratory to suspected virus must be grown on culture method known which support the growth of the particular viruses. So if we are saying in very broad line, for bacteriophage we require bacteria as a host. We do this practically in our classes. तो हम लोग बैक्टीरिया को यूज करते हैं बैक्टीरिया वायरसेस को ग्रो करने के लिए. एम्ब्रियो में यहाँ पे मेंशन है, we use a different parts of the embryo to cultivate the viruses. Then we are using mice. In the mice we have a specific location where we can grow our viruses. Then rabbit model we are also using. And lastly, nowadays the virus cultivation mostly goes on animal tissue cultures, right? Because they all are live. Apart from the bacteriophage, we are now focused on animal tissue culture for the culture because most of the cell lines now well established. So this is the better way to cultivate the virus, and for making the all types of most of the vaccines, we are using animal tissue culture right now. But what is the case of plant viruses, right? In the case of plant viruses, we maintain the virus into the live plant, right? If you got any plant material having the virus, you choose the selected or similar plant, that is a healthy plant, which here you find the my hand, which I maintain the above virus, that is a citrus pistosa virus. I work on a citrus pistosa virus. This is the RNA virus. Then citrus mosaic virus. So many viruses I work in a case of the plants. And I work on the corona also. So like this, we maintain the viruses inside the plant. This is the technique is known as biological engineer the virus through the symptoms analysis, right? I will tell you one thing that plant is always, I'm telling you the another slide. Okay? This one. See this one. If you are the plant viruses, and if I am talking about the citrus pistosa virus, right? This is the leaf of suppose any virus. If you find this leaf, on this you will thoroughly observe that it have the some vein clearing symptoms. Means this virus is a phloem limited. Hence these are the midrib and these are the some veins, right? These virus eat the whatever the nutrient present in that vein, he use that nutrient to survive themselves. Hence, in a winter season, if you find the acid lime plant, these plants, if appears that the vein clearing symptoms, means you are 100% sure that it is infected by citrus pistosa virus. And I will give you a guarantee that in 90% time I am correct to identify virus in this way. This is the particular symptoms given by the virus. This is symptoms analysis. On the basis of symptoms we can, just like a doctor, right? Then biological indexing, which method I told you this. If you done this all the task and then you put your samples into the plant tissue culture or the polyhouse, you require minimum 3 to 4 months to appear the symptoms on that particular indicator plants, राइट एक हेल्दी प्लांट के अंदर में हम क्या करते हैं, जो सस्पेक्टेड प्लांट है उसको हम ग्राफ्ट कर देते हैं जस्ट लाइक अ रूट स्टॉक सायन कॉम्बिनेशन में, राइट. फिर उसको हम रख देते हैं पॉली हाउस में कंट्रोल कंडीशंस में. अब क्या होता है, प्लांट क्या करेगा, प्लांट अपना रूट अपना सायन में बोले तो अपर साइड में है, सायन बोले तो दिस साइड में है, ये अपर साइड में रहता है, ठीक है. फिर वो रूट स्टॉक में जाता है और पूरा सिम्टम्स प्लांट में आता है. तो उसको लगता है कितना 3 to 4 months. तो बिफोर द डिस्कवरी ऑफ एंटीजन एंटीबॉडी, PCR, electron microscopy and all molecular biology technique, we require 3 to 4 months to detect the virus, right? Later on serological techniques comes. In these serological techniques, initially we require 1 to 2 days, जैसे एलाइजा बोलते हैं ना हम लोग, वो वाला 1 से 2 दिन रखते थे. देर आर सो मेनी एलाइजा टेक्निक जहाँ पे आपको रात भर एंटीजन या एंटीबॉडी को इंक्यूबेट करना पड़ता था आपके प्लेट्स के में ठीक होने के लिए. तो दीज आर द सीरोलॉजिकल टेक्निक्स आया. फिर 1 से 2 दिन में हम इलेक्ट्रॉन माइक्रोस्कोप में आए. तो इलेक्ट्रॉन माइक्रोस्कोप देखेंगे तो इसका भी एक अपना ओन लिमिटेशन है. इट रिक्वायर द ह्यूज लैब, skilled manpower, very costly instrument. If you are looking for the today's price also more than 40 to 50 lakh rupees. जब पहले आया था तब तो CRs में आता था ये वाला. Then skilled personnel रखता है इसको, liquid nitrogen, so many alternative things you require to run this electron microscopy, right? And from that half day, right, minimum required 4 to 6 hours while doing the OR processing, you require observe the microscope. फिर उसके बाद आया जमाना आपका मॉलिक्यूलर बायोलॉजी का टेक्निक का. जैसे पीसीआर का डिस्कवरी किया कैरीमुलिस ने, उसके बाद पीसीआर विल कम एंड पीसीआर आपके पास रेडीमेड डीएनए या आरएनए है, स्टिल इट रिक्वायर 3 घंटा पीसीआर करने के लिए. देन उसके बाद 1 घंटा इलेक्ट्रोफोरेसिस, और पहले डीएनए नहीं होगा तो 1-2 घंटा उसमें. तो स्टिल वी रिक्वायर अ कंप्लीट 1 day or minimum 6 to 8 hours to detect the PCR, right? That is a polymerase chain reaction. Later on we have the RT-PCR, right? This is very quick because we don't require electrophoresis here. You are getting the result automatically on your screen. It required again 2 to 3 hours, right? 1 hour at least for the preparations, and then runtime is also there. And if you don't have the nucleic acid isolated, then more
There is
time it will require.
there is one question.
Yes,
As
sir.
in animal virus, are cells or tissue specific? Does this same thing goes with plant virus also? Shubham
Sir,
will
yeah.
Bellow. Okay. आप देखिए.
Thank you for the questions. Means we have a very good audience, sir. Sir, like a plant viruses are the host specific. I am not going with the tissues, but they are the host specific. Means banana viruses is never in fact going to the citrus viruses, right? But yes, in a case of cucumber mosaic virus, if you see that the one virus has infect to more than almost 100 plus species of the plant cell. This is the tissue tropism. And in a case of plant viruses, there is a one study known as host range studies. We are doing so much studies कि ये वायरस उसको इन्फेक्ट करता है कि नहीं करता है. So yes, sir, some specificity shows by the viruses, but uh, some peculiar viruses can infect the so many host range like this. But one very peculiar funda I can tell you, animal viruses never infect plant viruses, right? Animal viruses never infect plant viruses, and plant virus never infect humans or animals. But nowadays, within these 5 to 3 years back, there are some reports not truly confirmed. Again, I am telling, the plant viruses is starting infecting humans also or animals also. Still, the data, a strong data is yet to come. Is it okay, Shubham? Gopal sir, shall I move?
Yes,
Yes,
sir.
yes,
Thank
yes.
you.
And
Yeah.
so you are discussing if virus is already known.
Yeah.
If some virus is novel virus, then PCR, RT-PCR will not work because for PCR sequence must be known. So suppose
Yeah, yeah.
first time it is reporting, then how we can uh, find out
Sir,
whether it is viral
yeah,
disease or bacterial or something else.
very good answer, sir. Sir, first time you know, sir, coach postulates, right? There are the four coach postulates. If you have the known disease, yes, it is a disease. We have to first establish that it is disease or not, sir, right? For that, all coach postulate it should follow, right? Once, if any case of the disease, there is involvement of the pathogen, right, sir? Second postulate, you have to isolate it. So if the virus is there, it can be isolated and we can culture into the some, you can medium, right? The virus here is not culturable, right? Again, we can transfer this virus via rupturing to the leaves, then sap को transfer करके या फिर कोई stem को biological indexing करके वो दूसरे प्लांट में वो ट्रांसफर होता है कि नहीं और वो प्लांट वायरस में मेंटेन होता है कि नहीं ये भी देखना है सर हमको फिर से आइसोलेट करके उसको फिर से रीइंफेक्ट करना है तो ऑल फोर कोच पॉस्टुलेट वो ओबे करता है then it is confirmed that it is a disease sir means there is involvement of pathogen now if you want to identify the virus so now we have the next generation sequencing sir right a complete DNA sequence we can do and nowadays sir with the high throughput sequencing this work is already going on into the IRI Indian Agriculture Research Institute we are sir now identifying very new viruses also present in a plants also so this way you can identify the viruses in that case which is you don't know the genetic material of the that this is the one thing sir
So only one way sequencing is only way to.
yes currently sir currently this is the method which is able to identify or otherwise some random sequencing or random primers were used sir sometime once we got the band then we are doing the sequencing of that apart from that still don't
Means
have the
we
technique
have some sets of primer specific to virus. Are you
yeah
saying? Okay.
yeah, yeah.
Yes.
Mostly for the detection purpose sir mostly for diagnostic person the researcher or the scientist and even me also are using we are using specific primers okay of
That's means it's known. Then a
it's
specific
known
primer.
yes sir
Otherwise, it's not known.
अदरवाइज
Then
वी डोंट नो बहुत बार सर क्रॉस मैच कर दे अभी मैं आपको और एक चीज बता देता हूँ ये बहुत क्वेश्चन है इफ यू वांट टू डिटेक्ट द वायरस एनीवन वांट टू डू स्टडी ऑन द वायरस design the primer for its code protein gene right? Just like when we have to identify or characterize the bacteria, we are using 16S rRNA. But when you are using the virus, in a case of virus, you can use code protein gene, right? In the case of bacteria, 16S RNA. In a case of fungi, IPS region, right? In a case of human, go with the VNTRs or the 18S RNA, right? Or 23S. In a fungi, it is the 18S ITS region, right? So these are the particular sequences or the regions in a genome which we can target for the detection and identification.
Okay.
In a case of
So,
virus, directly go with
so,
the code.
so like in bacteria, 16S ribosomal genes are conserved almost, है ना?
Yes, yes, sir.
Same way for capsid, it's for all virus that gene is conserved.
Not all virus, sir. It is specific to specific. There is no single gene present in the viruses which is conserved, sir.
Then it's difficult.
It,
Na?
hence, sir, people are very less people are working on viruses, sir. This is the main reason, and hence it is a very different from others also. Getting, sir?
Okay, okay. Please continue.
इसीलिए थोड़ा टफ है सर वायरोलॉजी में काम करना, but still people are working, sir. So shall I move?
Yes, yes, yes.
Okay. तो फिर उसके बाद RT-PCR है. This is the all my research papers are available on this all techniques, including biological indexing, serological, all molecular like RPA, all I prepared in my labs. We publish the protocols. We have our own kits. Now, RT-PCR बोलते हैं रियल टाइम PCR, जिसमें आपको मिनिमम 3 से 4 घंटा लगेगा ही लगेगा. Then we develop in the first time India we developed the LAMP technology, but this technology is from the abroad, right? Alkane का उसका technology था, but for our viruses we use in a LAMP technology. LAMP means what? I'm coming later on, right? Then RPA technology, that is a recombinant polymerase amplification. अब ये जो लैम्प टेक्नोलॉजी है, विद इन दिस टेक्नोलॉजी वी कैन डिटेक्ट द वायरस विद इन 2 hours, and this RPA technology we can detect the virus in a 40 minutes, right? And now some lateral flow is available that is based on antigen antibody, जो viruses highly immunogenic रहते हैं, मीन्स किसी एनिमल होस्ट में डालेंगे तो अच्छे से एंटीबॉडी प्रोड्यूस करते हैं. We can detect within the 5 minutes using the lateral flow assay, right? So we are coming on this technology one by one. So this is the all biological indexing, nothing but symptoms analysis. This is your ELISA. This we are missed, that is the radio level probes. Now no one is using this. Before ELISA और PCR के बीच में ये टेक्नोलॉजी आई थी, रेडियो लेवल प्रोब्स. जब हम ये करते कॉलोनी हाइब्रिडाइजेशन करते थे, वो वाला टेक्नोलॉजी था ये वाला. Then we have PCR machine, right? जो आपको almost 4 से 6 घंटे में दे देगा. Then we have real time PCR 3 to 4 hours. Then we have LAMP which can give result in a hardly 1 to 2 hours. And then we have the lateral flow assay. It can give the result within the 5 minutes to 40 minutes. This is the power of the biotechnology basically, right? Using this biotechnology we develop such kind of the technology. These all are my figures, okay? Then we will go later on. So detection of viruses to PCR we will see. So कैरीमुलिस जब उसने PCR इन्वेंट किया तो इसने पूरा बायोटेक्नोलॉजी को ही चेंज कर दिया, और वहीं से जन्म हुआ एक नया ब्रांच का, दैट इज नोन एज बायोइन्फॉर्मेटिक्स आल्सो, राइट? So it is the most revolutionary method he developed, that is the कैरीमुलिस, best invention of that era, based on now the, if कोई सिंगल वर्ड में आपको पूछता है कि व्हाट इज पीसीआर, तो सिंपल बोलना है. पीसीआर का जो प्रिंसिपल है वो है डीएनए रेप्लिकेशन. व्हाट इज द प्रिंसिपल ऑफ पीसीआर? डीएनए रेप्लिकेशन. व्हाट इज द एप्लीकेशन ऑफ पीसीआर? इज द डीएनए एम्प्लीफिकेशन. दिस टू वर्ड्स ओनली यू कैन डिस्क्राइब द पीसीआर. जो पूरा डीएनए रेप्लिकेशन हमारे बॉडी में होता है उसको हम पीसीआर में बाय हीट से करते हैं, टेम्परेचर से करते हैं. Then used for the detection of any kind of the pathogen. If आपका पैथोजन न्यूक्लिक एसिड जिसमें है उसको हम पीसीआर से डिटेक्ट कर सकते हैं. Then here primers decide the specificity of virus. Required purified nucleic acid. In a case of PCR you require isolated, extracted, purified DNA. This is the limitation of your PCR basically. This is not a limitation in the case of LAMP or RPA. Then you find that my students in my laboratory, they are developing the PCR based assay for the plant viruses. Then as I work on the citrus viruses, by morphologically, okay, by naked eyes or by symptoms analysis, you find that this is the not good citrus plants basically. आप इसको देखेंगे तो इसमें स्पार्स फोलिएज है। स्पार्स फोलिएज बोले तो जहां से आपको जिस प्लांट से सीधा सन दिख जाए, तो समझ लेना कि इट इज इन्फेक्टेड बाय पर्टिकुलर वायरस, दैट इज सीटीवी। और ये जो संतरे का जो वायरस होता है दैट इज नंबर 1 वायरस, और ये सबसे बड़ा वायरस है। 19.2 केबी इसका जीनोम है। इसका मॉर्फोलॉजिकल साइज भी बहुत बड़ा है, दैट इज सिट्रस ट्रिस्टिजा वायरस। देन उसका इलेक्ट्रॉन माइक्रोग्राफ हमने लिए थे, दिस वन। Then you find these are the PCR bands which we develop in our lab only, right? So this is it is a RNA virus. We develop the RT-PCR for that. It is showing the perfect band around 672 base pair, means it is a positive one. We also cross check with the sequencing also, and then we have the create a duplex PCR. Duplex means a single PCR we can identify the two different viruses together using only two different set of the primers. We develop in our lab which can detect the two virus at a time. Here we can save the time, money, consumable, manpower also. And we can give the result to the users basically or our customers. This is the case. Then, एक second. सर एक लेक्चर ले रहे ऑनलाइन, आप 10-15-20 मिनट में। then you find that the real time PCR, right? So this is the machine which we have, which I work on this machine. So this is the real time PCR. Then it is a gold standard detection technology right now we are using, right? This is the most gold standard because it having the specificity basically for the Tachman assay, not for the cyber green, routinely used for the detection of coronavirus. And now most of the detection kits is based on real-time PCR. It gives the result very sensitivity be high or uska specificity be high. The result obtained in a real time, that is you observe on the screen based on two chemistry, that is the cyber green and Tachman assay. Now what is the difference with cyber green? Is a dye basically which can bind into the two strand of the DNA, whereas Tachman is based on the probes, additional. Apart from the primer, we are using the probes here, which is the additional supported for this. So that is the Tachman assay which we developed here. Then this is the one technology that is known as LAMP. LAMP means what? Loop mediated isothermal amplification, right? So this technology, you don't require any kind of sophisticated instrument. In simple water bath or simple dry bath, you run this reaction and you will get the result. So it is work on a strand displacement and auto cycling activity, BST DNA polymerase. It is a very good DNA polymerase which works on strand displacement activity. You get very good amount of DNA here. PCR में हम 2 primers use करते हैं, लेकिन यहाँ पे 4 to 6 primers हमें use करना पड़ता है. Isothermal means it require only one temperature, either 60 to 65 के बीच में काम करता है. But PCR में देखेंगे आप annealing, denaturation, again extension is there, but such case is not available with this. Then LAMP is very sensitive, simple, robust, low cost diagnostic technique which developed. We amplify the DNA within 1.5 hours if we have the ready-made DNA with us. No need of any sophisticated instrument. This is the first report which we developed the RT LAMP assay for detection of CTV from India. Like this, so many viruses, first time we detect through the LAMP protocol. We publish in a very good journal like Elsewhere, Springer's. And this is the one of the way to do the research also to detect the virus or detect any pathogen in minimum cost for the humanity or for our society. So this is the LAMP which we publish. So this is the plant, citrus plant infected with CTV. We have maintained our viral cultures in our own polyhouse, which showing the symptoms like this. And then we develop this assay. All this green color. Before reaction, we all have this violet color, right? After reaction also have the violet color. but one additional dye we added into the all tubes, that is cyber green dye. If addition of the cyber green dye, if the color is turned violet to green, means it is positive. These four are positive, whereas these three are negative. When we run a electrophoresis, that is additional confirmation we are giving, we get a ladder like band. If you see these all four tubes, you are getting the ladder like band, means it is positive, others are negative. Then this study which we done, we also identify to the PCR. Same same samples we run with the its primer, specific primers using the code protein. We are getting positive only in the four. Then we are using the outer primer of the same lamp. We are getting the same result. This confirms that our technology detect the viruses just like a PCR and real-time PCR, and its sensitivity is very like this. The only problem with this technology, as it produces a very high amount of DNA, there is a many chances to cross contaminations or aerosol contamination. In this case, if you taking care of this one, otherwise this technique is a good. Then we come to the recombinant polymerase assay. That is the LAMP RPA technology. In a simple suitcase, you can detect this virus within the field. In the case of plant viruses, तो आपको ये जो दिख रहा है, ये टेक्नोलॉजी काम करता है. अगेन आइसोथर्मल है, पोटेंशियल अल्टरनेटिव. इसके बाद कोई पीसीआर को रिप्लेस करेगा तो वो आरपीए रिप्लेस करेगा, ठीक है ना? Because both technology has a nucleic acid base. By adding the reverse transcriptase, you can do the RT-PCR also. Again, it requires the water bath simple. It can be used as a point of care technology because you can detect in a field also. Within hardly 40 minutes is required to take the result from this. And again, just like a pregnancy kit, you will get the result on the this lateral flow. So we also develop this technology for Candidatus Liberibacter asiaticus. We published that paper into the PLOS one also. You can visit my Google Scholar, so you will get this all paper there. Then now we come to the bacteriophage. That number, the bacteria which infect the, sorry, the virus which infect the bacteria, it is known as bacteriophage, and 10 raised to the power 31 bacteriophage, which I told you earlier. This is the number we have of the only case of bacteriophage. Then you find this image. अब तक हम वायरस को क्या बोलते थे कि वो गंदा है. वायरस इज वेरी प्रॉब्लमेटिक, वायरस बहुत कुछ करता है. बट अभी आप देखेंगे तो देर इज अ वन प्रॉब्लम इज एंटायर वर्ल्ड इज फेसिंग, दैट इज एएमआर, एंटीबायोटिक resistant, right? So many antibiotic now become resistant. So many bacteria got resistant for the antibiotics. Sorry for that. So no one bacteria is going to kill by the antibiotic. If you are looking for the TB, you require 8 antibiotics to kill TB, right? With so many antibiotics now become resistant. But now what is the solution we have? You know that bacteria can infect, so viruses can infect the bacteria. So now we use bacteria, viruses to kill the particular bacteria in the form of bacteriophages. So initially what antibiotic tell, I will kill all of you, right? But now bacteriophage can tell that I will only kill you only. Means particular बैक्टीरिया को, पर्टिकुलर इन्फेक्शन को हम उसका रिस्पेक्टिव वायरस से उसको किल कर सकते हैं. इट इज नोन एज बैक्टीरियोफेज थेरेपी. It is now applied to the so many sciences in animal science and plant science also. Then replication is always from the lytic cycle and lysogenic cycle. ठीक है? I'm asking one question to all of you that एक बैक्टीरिया को जब एक वायरस इन्फेक्ट करता है, राइट, तो कौन डिसाइड करेगा? Who will decide that inside the bacteria it is goes to the lytic cycle or lysogenic cycle, right? आप सबने पढ़ा है ना lytic cycle, lysogenic cycle?
Yes, sir.
So do you know the answer of this question?
Lambda repressor and co-repressor concentration.
Concentrations. Okay, you are very close to answer, but you will try to find out the answer and later on you told me, right? You are very close to the answer. Okay, so we are moving now. So these are the symptoms of Tristeza virus, right? So if you see, this is the midrib and this is the their veins. It is showing completely clear. And in a deep picture, you find that this is the healthy leaf, right? This is the healthy leaf, and you find a particular symptoms in a case of plant. This is vein clearing symptoms. This is leaf yellowing symptoms. This is again midrib yellowing symptoms, right? If you got such symptoms, then you can say that yes, it is a virus. Then you will get the midrib, okay, very rough midrib you are getting, that is a vein yellowing symptoms. Once you got into the field, you can come to the conclusion that this is the symptoms analysis in a case of CTV. Then beautiful symptoms you will get here. You are eating this papaya mostly, right? That is a papaya ring spot virus. You can eat very safely because papaya ring spot is not going to infect you. Okay, it only found most of the times on the symptoms appear on the upper walls of the fruits, right? So you can consume this fruit, but farmers will get a very lesser price for that. Hence, now the work is also going on this also. Then soybean yellow mosaic virus. This is from last 3 years is the most devastating virus who can destroy the so many crops in Maharashtra and Madhya Pradesh and many parts of the India is very devastating. जहाँ पे 10 क्विंटल सोयाबीन पर एकड़ होता था, वहाँ पे अभी 2 to 3 kg भी नहीं हो रहा है, थ्रू क्विंटल भी नहीं हो रहा है. This is the problem with this virus, and this leads to the research, right? This is the TMV. Again, this is the Indian citrus ring spot virus. It is again limited to the leaves only. That is not so much devastating one, but due to appearance of such symptoms, problem is going on. But here in the case of cucumber mosaic virus, so you find that this is infected by virus, and this is normal one. This kind of cucumber is no one going to purchase in a market, right? So farmer facing a lot of problems regarding this. So we have to work to solve this problem, right? Then again greening disease. Again, it is showing a very particular symptoms. If I am an expert in this field and I got the symptoms like this known as green Iceland symptoms, means you find the yellow leaf on that small green island is there, then you are 100% sure that it is a citrus greening disease. Again caused by, it is a bacteria but behave like a virus. Till date no one culture it. पहले तो इसको लोग वायरस ही बोलते थे, बट जब सिक्वेंसिंग हुआ इसका तब जाके पता चला कि एक बैक्टीरिया, और आज भी अनकल्चरेबल सारी प्रॉपर्टी इसका न्यूक्लिक एसिड छोड़ के सारी प्रॉपर्टीज वायरसेस. दिस इज द प्रॉब्लम. And once it is infected, this will come, and within 10 years of the span or 5 years of the span, this plant is going to collapse. It is not going to bearing the fruits. This is the case in a case of citrus greening disease. Then you find some important viral diseases.
How many slides left?
So only 2 to 3 slides, sir, not more
Okay,
than that.
okay, jaldi.
So then viral diseases you find that chikungunya, poliomyelitis, rubella, hepatitis, chikungunya, AIDS. जितना भी ये वायरसेस मैंने आपको स्लाइड दिखा रहे हैं, ये मोस्ट ऑफ द 90% आर आरएनए वायरसेस, and RNA viruses are more devastating, more dangerous, with very less control by the humans. So handling with the RNA viruses is more difficult than DNA viruses in practical also. In a case in a field, they are more devastating for the humans, plants, and others also. So we have now come up with this antiviral agent which can target the virus in their particular replication area. So now this is the very good area to do the research how to control the viruses in this area. So this is the task we have to take now, and this is the target which I gave. On this target, if we attack, we can able to control the virus in this manner also. So if now the emerging diseases, आपको जितना भी रेड डॉट दिख रहा है ना यहाँ पे पूरा ग्लोब में, ये री इमर्जिंग और इमर्जिंग हो रहा है. सार्स पहले 100 साल पहले आ चुका था, फिर से आया अपने पास, राइट? So all these are the upcoming new challenges. Coronavirus jab aaya na, this is not a last. It is only starting. You are facing one by one. अभी recently आ रहे हैं ना बहुत सारे syncytial virus, pneumonia. तो ये तो अभी शुरुआत है. अभी धीरे धीरे आपको you will get the more viruses very soon. So these are the beautiful pictures you will have to see. These are the smallpox, bird flu, herpes virus. And our research is what we done in last 12 years from my PhD to up to now. We publish good papers. We publish a good product which can control the viruses or virus-like organisms. We got some award and our research is published into the daily newspapers like Times of India, which is with the IIT Roorkee. This work where the lab which I work with the IIT Roorkee, ICR, Central Citrus Station Nagpur, and University of Florida. Dr. Santra is there. And for that, we also got the Young Scientist Award. Then, this is the my team, which currently I work with this one. These are my desert station student and my project student as the JRF and SRF they are working. These are my collaborator, Dr. Ghosh is there, who is director in ICR. Then Dr. Chopre, who is the vice chancellor of AKS Satna University, and Dr. Rashmi with the Delhi University. She's the Jigisha, Yash, and Isha. Again, the three members which joined us very recently, and this is the lab which we have currently. So currently I'm working in a Gujarat Technological University, in which our department is a School of Applied Sciences and Technology. Dr. Gopal is recently visited our university, and this is the lab which we have, and our research scholars are working in this laboratory, and we are working currently on the natural farming, then citrus viruses, banana viruses, and currently we are working on the fungi also. We started. So thank you very much for your kind patience. Thank you very much for the Gopal sir who invited me for this talk. I'm very happy to deliver the lecture for the all budding scientists in the future. For question, I'm most
Thank
welcome,
you
sir.
very much. It was really so many things we have learned, and obviously, then other participants have learned so many things. If
Thank you, sir. Thank
anyone
you.
have any question, please ask. But we have not stopped you till last slide to ask question because we have changed the pattern. Generally, in webinar, people ask question after final slides, but here
Right.
we allow our participants to ask question anytime. But
So
still,
you know
we have
now, for our lecture, we require minimum 3 hours to do this virology, right?
yes, yes,
But we
yes.
try to come up with the 1 hour.
Yes, you can unmute and ask. Susant is asking, sir, how can join with? So you can qualify that PhD exam of GTU, I think, Hannah.
Yeah, yeah. I am a
How
PhD
you
guide
can
over here.
join with project next project.
Sir, I am a
Sir,
PhD guide over here. You can join as a PhD student with me. Or I have couple of projects which is going on. Currently, all vacancies are full. But if the project is come, you can join with me, or as a trainee, you can join with me also through proper channel.
yes, sir.
Yes.
Good afternoon, sir.
Thank you, sir.
Sorry, I'm Dr. Vipul Patel,
Yeah.
and it's a wonderful lecture session, sir.
Thank you.
I'm working as a head of the Department of Pharmaceutical Biotechnology,
Yeah.
and would like to request for project intern on our at your institute because it's
Yeah.
having a very good sophisticated lab and a very good exposure. So if
Right.
we are getting chance, let us know in which particular phase we are getting chance to have a project intern over there from our side, sir. PG student, like, yeah.
Okay, you want to send your dissertation student or PG student to work in our laboratory, right, sir?
Dissertation, yeah, interns or project training, like
Yeah, right.
whatever is it
What
possible.
you can,
Yeah,
you can do what you can do. You
yeah.
can write either mail to me, sir, my mail with the Dr. Gopal,
Okay,
right? Or you can write the
okay,
mail to the my director, sir, requesting them that you want to do
okay,
these things. Sir, we have the 11
okay,
faculties at our SAS, at our school, all having the different expertise. Okay. So if you find that my profile is suitable for your dissertation student, they can
okay,
approach me through the mail to me or my director, right? If they approach to me, then I will have to take the permission from the director, or then you write the mail to the director, and the director will take the decision, and he can allow something. So
okay,
this is the way.
okay. Thank you so
The
much for
first time,
wonderful, and thanks a lot, Gopalji, sir. You're really very inspiring these things, and a good initiation. We are getting much benefited for that. Thanks, sir. Yeah,
थैंक यू सर थैंक यू
thank
मैं
you.
देखता हूँ आप
yeah.
बोलते हैं कि लिंक नहीं एक्टिव हो रहा है लिंक आ रहा है ना. So I
Yes,
always
yes,
try
yes.
as you
Yeah,
know,
yeah, yeah.
मेरा जन्मभूमि तो बिहार है लेकिन कर्मभूमि गुजरात है, so I'm trying to
Yes, sir.
uplift overall, है ना। अभी
Yes, yes,
अब मैं
really good.
5:30 से जीनोमिक्स प्रोटियोमिक्स कोर्स कर लूंगा, और 2:30
Okay.
से 4 बजे तक, पौने 4 बजे तक मैंने लिया था एनएमआर, और उसका जो है ना, मैंने मतलब
Yes,
हमारे
yes.
उसने है ना सेमिनार का,
Yeah, yes, sir.
and then we
Really
are
good,
going
sir.
to conduct 15 days offline course for BSc final year student, and everything is that cost will be aired by GSBTM. 15
Okay, but
days.
unfortunately, sir, we are from other state. I am from Maharashtra. I belong from Gujarat, but I am giving my service to Maharashtra right now.
Good.
Okay, okay. Anyway,
Yes,
it's a good thing,
yes,
sir.
It is a vice versa, sir. Vipul sir, it is a vice versa basically. I am
Yeah,
from
yes, sir.
Maharashtra,
Of course.
yes.
serving in Gujarat.
Yes, we
Yes,
should
But
yes,
the
yes,
collaborate.
yes.
आजकल तो
policy over
ऑनलाइन में सब प्लेटफॉर्म पूरा ग्लोब ही हो गया है, कंप्यूटर में सिमट गया पूरा
Yes,
ग्लोब।
yes, yes. Nothing to worry about border and all like.
यस, एंड
Thank
ये
you, sir.
लेक्चर कराने के ये भी सर है कि एक्सपर्टाइज लोग एक दूसरे का जान पाएंगे, सो इन फ्यूचर दे कैन
Yes, yes,
कोलैबोरेट,
sir.
दे कैन टेक देयर हेल्प, है ना। So
Yes, definitely, sir.
yes sir, any other participants have any question or suggestions or feedback?
okay. वो जो
We,
क्वेश्चन जो पूछा था ना बित्रा, बेटा
हाँ,
इट इज ऑन
Yes,
इट्स
sir.
प्रोटीन, क्रो प्रोटीन। आप है ना वॉटसन, मॉलिकुलर बायोलॉजी बाय वॉटसन पढ़ना, उसमें उसका पूरा मैकेनिज्म दिया। इट इज बेस्ड ऑन द इट्स क्रो प्रोटीन, राइट। एक सिंपल सा आंसर बताओ, इफ आपके घर में खाना वाना बहुत अच्छे से है तो आप किसी गेस्ट को अलाउड करेंगे, राइट। इफ बैक्टीरिया के पास वेल फूड है, उसके पास खाने के लिए अच्छा है, तो देन इट विल गोज फॉर द लाइटिक साइकिल, राइट। इफ उसके पास कुछ भी नहीं खाने के लिए अभी, तो उसमें आएगा और उसको डॉर्मन कंडीशंस में रखेगा, ठीक है, तो वो लाइसोजेनिक साइकिल में जाएगा। बट आप मैकेनिज्म के थ्रू जाएंगे, you will get the two protein, that is the pro protein, which is present in the, you go with the Watson, you will get the detailed answer of that. But simple theory is like that, you have a pro protein in that,
Sir, sir,
right
sir.
Mitra?
Uh,
Okay, sir. Thank you, sir.
Sir, I had one question. Suppose I am doing one formulation for anti-HIV, sir, and I need a facility of viral testing, if possible, over there, because NIV and NARI and all I have tried many lots time, but they are not allowed because of their busy schedule and all. So, is it possible over there?
Sir, actually animal tissue culture facilities are handling by the other professors, but what I
Okay.
understand, we require a host cell line for that,
Ah,
right?
that's okay. That will
Okay,
bring it down. And
yeah.
BSL-3 चाहिए होगा ना।
हाँ, वो मेरे पास शायद नहीं है, कुछ वायरस हम बैक्टीरिया पे जनरली काम करते हैं, ठीक है
हाँ,
सर। बट इन दैट केस you have right to the particular person, but such facility is not available with us most probably, right?
विपुल
okay.
सर,
हाँ,
GBRC आप ट्राई कर
हाँ,
सकते हो ना,
GPR से आप ट्राई कर सकते
okay, sir.
GBRC में होगा, और वहाँ फैसिलिटी
अच्छा,
भी है थोड़ा अच्छा सा।
हैं। Very
रोटेटा,
soon they
okay.
will have BSL-4 also, very soon.
यस, और
Okay.
वहाँ पर में आप जो है ना, बिना कोलैबोरेशन का भी यू कैन पे, दे आर
Ah,
चार्जिंग
that is
यू, है
okay.
ना।
That's good thing, na, sir.
So
If we
हाँ,
are getting that
this is
facility.
government funded, so charge is also very less. Anybody can
Okay,
utilize that resource. Gujarat
okay,
Biotech Research Center, है ना। I
okay,
have
sir.
even sir
visited.
मैं भी आपको हेल्प कर पाएंगे, we have a collaboration with the GPRC,
Ah,
right?
that's
Yes,
a
yes.
TBZ cell line is there, and we are controlling with the exposure and all. Okay,
I
sir.
will tell
Thank
you
you.
sir how to proceed with it.
फाइजा क्या क्वेश्चन है चिकनगुनिया का ट्रीटमेंट? शी इज आस्किंग चिकनगुनिया का
Well,
ट्रीटमेंट।
till date we don't have chikungunya, right?
So
We don't
आप,
have any antiviral for the any viruses. हमारे पास जो भी अभी immune booster है सारा, ठीक है? Till date we don't have any antiviral molecule like in corona also, right? आज तो कोई proven नहीं है। आज आप उसको है ना antiviral करा दोगे, next दिन ही जाके वो resistant हो जाएगा उसके लिए, right? बट मेरे लिए आपके लिए एक बहुत अच्छा क्वेश्चन है, आप घर में जाके ब्रेनस्टॉर्मिंग करना। व्हाई चिकनगुनिया वायरस कॉल्ड एज चिकनगुनिया, ठीक है? फिजा, आप एक क्वेश्चन करना और मुझे आप
Yes,
पर्सनली
sir.
आंसर कर सकते हो। व्हाई चिकनगुनिया वायरस इज नोन एज चिकनगुनिया, इज वेरी फैंटास्टिक आंसर।
yes, Joshi Dhruv P is saying RNA silencing is the viral remedy. Yes.
बट इट इज नॉट इन मार्केट, इट इज ओनली विद द रिसर्च पेपर, नॉट टिल इन अ मार्केट, राइट? अभी नहीं है हमारे पास जीन थेरेपी भी, उतना स्ट्रॉन्ग आंसर नहीं है हमारे पास। हम ये सब पेपर्स में देखते हैं, राइट? and that
पार्थ
paper is
जोशी जी
again is
बोल रहे हैं या
not
जोशी ध्रुव?
हाँ yes sir
नहीं, ये मैं जोशी ध्रुव देख रहा हूँ, एक पार्थ जोशी भी ज्वाइन किए थे।
okay
ए,
okay
वो भी हमारे रिसोर्स
but
पर्सन
RNA
रहे।
silencing is not come into the market till date no strong antiviral was about the aclovir. These are the drug which can inhibit the virus, not completely kill the virus. Not completely kill. It is only inhibit the replication of the virus. They are not killing the virus.
विद्यानंद झा।
Yes. Sir, we are getting a very good response. Means my
Yes.
student understand the lectures. So I'm very
All
happy for
viruses
it.
have antigenic shift and drift? Question mark.
Yeah, they have. They are doing so much mutations. Basically, ये जो मैं बोला ना अभी कोई वायरस HIV के लिए कोई वैक्सीन आया नहीं अभी तक. You know why? It changed its receptor. इतना साल हो गए लेकिन हमें उसके लिए वैक्सीन नहीं मिला. पता है आपको क्यों? बिकॉज हमारे बॉडी में जब डीएनए रेप्लिकेशन होता है या बैक्टीरिया में डीएनए रेप्लिकेशन होता है तो उसको ठीक इफ यू हैव अ प्रॉब्लम विद डीएनए रेप्लिकेशन वी हैव रिपेयर सिस्टम राइट बट वी हैव द प्रूफ रीडिंग सिस्टम विद आवर पॉलीमरेज बट ये प्रूफ रीडिंग सिस्टम एचआईवी वायरस में है ही नहीं राइट उसका रिवर्स ट्रांसक्रिप्शन प्रूफ रीडिंग सिस्टम है ही नहीं इसीलिए वो वायरस को बहुत म्यूटेशन करता है और वो अपने रिसेप्टर साइट चेंज करता है दैट्स व्हाई वी डोंट हैव एनी स्ट्रॉन्ग एंटीवायरल एजेंट और वैक्सीन अगेंस्ट एचआईवी उसको डिटेक्ट हुए ऑलमोस्ट 30 साल हो चुके हैं वी डोंट हैव अ सॉल्यूशन
Okay. So now, yes, Parth Joshi now he has asked. Evolutionary conserved. Was there any source virus from where? Or
yes it is coming there are three yes part there are three different stories from where evolution come first one it is जैसे उसका होस्ट का इवोल्यूशन हुआ जैसे संतरे का इवोल्यूशन हुआ तो उसका सिट्रस डिसीज वायरस का भी इवोल्यूशन साथ में हुआ ठीक है दिस इज द फर्स्ट स्टोरी देन क्या हुआ बहुत सारा जब बैक्टीरिया रेप्लिकेट हो गया थोड़ा सा जीन बाहर पड़ गया इस थोड़ा सा डीएनए डिग्रेडेशन होके जीन बाहर पड़ा उसी के अंदर में थोड़ा सा कैप्सिड हुआ और वहां से वायरस का इवोल्यूशन हुआ राइट और हमारे बॉडी में बहुत सारा ब्रेकिंग ऑफ डीएनए होता है ना देयर आर सम जीन्स आर वर्क टुगेदर एक साथ जीन जुड़े और उसके ऊपर में प्रोटीन कोड आ गया और वहीं से वायरस का इवोल्यूशन हुआ तो दीज आर द थ्री डिफरेंट स्टोरीज रिगार्डिंग द इवोल्यूशन ऑफ वायरस
अभी तक इवोल्यूशन का जितना थ्योरी है सब पार्शियल, किसी का
सब
बहुत
पार्शियल
ऑथेंटिसिटी नहीं है।
yes
पार्शियली
it's true
जिसको जो मिलता है दे गिव सम थ्योरी, है ना, सम हाइपोथेसिस।
hence it is known as theory not a
Yes,
proof
yes. So thank you very much, all the participants, our resource person, for joining us and disseminating his knowledge to wider audience, wider research scholar, life science researchers and scientists, so that knowledge pool will increase. हमारा जितना जो population है वो कारगर होगा, ये हमारा ध्येय है. So that people will come. So bioscience subject becomes more lucrative, more attractive, more fruitful, more job oriented. That is purpose ultimately, because people are living. जबकि सबसे important subject यही है, जीवन का विज्ञान सबसे important है, but people are joining some MBA, some CA, some this thing, that thing, because they get more salary. You all should know about life science, है ना? You all should join and spread the knowledge of life science, because life is very important and we all are trying to improve it. Yes, ठीक है,
ठीक है सर
ठीक है. Thank you very much.